Imagine standing at the entrance of a simple, nondescript cave. Not a dramatic site with ancient tools, cave paintings, or human bones. Just layers of undisturbed soil that seem insignificant to the untrained eye. But in the right hands, every grain of soil becomes a witness to human history. Hidden within that dirt are genetic echoes of the past, tiny remnants of ancient lives that can reshape our understanding of where we came from and who we are. What if the most important fossil isn't a bone, but a molecule? This is the world of sedimentary DNA, a field that recovers what can't be seen, genetic traces that sank into the earth after ancient humans passed through. Recovering that DNA can feel like searching for a single sentence in a book from a library buried under sand, but that one sentence might rewrite entire chapters of human history. Unlocking these hidden chapters requires cutting-edge lab technology techniques that can extract, purify, and read the faintest genetic signals from grains of soil. Join us on the journey of sedimentary DNA, from the cave floor to the lab bench to the digital world of bioinformatics. Step 1. Choose the best site. Scientists look for caves or other sheltered environments where early humans may have lived, rested, or traversed. Ideal locations are protected from wind, rain, and sunlight, and they have deep, stratified sediment layers. These places are often rich in archaeological remains or lie along known migration routes. Once inside the cave, scientists must decide where to sample. That's no easy task. It's difficult to predict exactly where any ancient DNA will be preserved, and ancient human DNA is certainly no exception. So they target specific sediment layers that correspond to different time periods, allowing them to focus on the eras when humans were most likely present. They're not just digging in the dirt, they're unearthing identity. These layers, often distinguished by subtle changes in color, composition, or texture, act like time capsules, preserving traces of human activity across time. But finding the right layer is only part of the challenge. Scientists must also prevent contamination from modern DNA. To avoid introducing their own genetic material into the samples, scientists must use sterile tools, gloves, and sometimes even full-body protective suits. Samples are then sealed and kept in cold storage to preserve the fragile molecules. Finally, a few grams of soil, painstakingly chosen and carefully protected, can begin their long journey to the lab, where they may at last reveal their ancient secrets. The stakes are high, because finding even a few preserved strands of ancient DNA can reveal forgotten migrations, lost populations, or unexpected connections between distant groups. Sometimes, a teaspoon of soil can overturn decades of assumptions. After transport, the sample arrives at the clean lab, a sterile lab environment free of all pesky contamination. Here, the real detective work begins. This sample may look like plain dirt, but hidden inside are microscopic fragments of ancient DNA. To reveal them, scientists treat the sample with special chemicals that release any DNA still clinging to the sediment after tens of thousands of years. These DNA fragments are incredibly small and exist in extremely low concentrations. Extracting them requires highly sensitive techniques, precision, and extreme care, preserving every molecule that might hold a clue to our distant past. Once the DNA has been extracted, it is converted into a DNA library, an organized set of DNA fragments that scientists can study. Scientists attach small artificial DNA fragments to each and every DNA molecule in their DNA extract, allowing them to copy and sequence these molecules later. Next, enter PCR, the polymerase chain reaction. Now, you may remember PCR from the COVID pandemic, when everyone from scientists to your grandma was trying to get their hands on a PCR test. But even though PCR became an overnight celebrity thanks to COVID, you likely have no idea what PCR actually is. Think of it as a molecular photocopier. It takes those tiny DNA fragments and creates millions of identical copies. In COVID testing, PCR was used to amplify traces of viral genetic material, so it could be detected, even in tiny amounts. In ancient DNA research, PCR does the same job, copying tiny fragments enough to actually sequence and study them. This is crucial because ancient DNA fragments are often too small to analyze directly. PCR also creates an immortalized archive of DNA, as a DNA library can be copied again and again and again as much as needed. At this stage, scientists are left with a diverse mix of DNA, including but certainly not limited to human DNA. The sample contains genetic material from all sorts of organisms: animals, plants, bacteria, and fungi. Sometimes, up to 90% of sediment DNA can't be identified, as it doesn't match anything in existing databases, or even used due to being degraded. So, how do scientists separate the tiny traces of archaic human DNA buried in that genetic jumble? This is where a method called hybridization capture comes in. Scientists take tiny artificial DNA sequences, known as baits, that specifically bind to the DNA sequences they're looking for. These baits are made using known genetic material, such as sequences from Neanderthals or Denisovans. Because single-stranded DNA naturally sticks to its complementary strand, the baits can fish out matching fragments from the chaos. It's like using a custom filter to sort through a mixture. Only the pieces with the right structure can break through. In this case, only the human DNA is extracted. Essentially, hybridization capture helps isolate the relevant fragments from the background noise. Now comes the moment when molecules turn into sequences. Scientists use a device called a sequencer. Think of it like a high-tech barcode scanner. But instead of scanning prices, it reads the genetic code, letter by letter. The physical DNA fragments are translated into digital strings made up of the 4 genetic bases: A, T, C, and G. Most modern labs use a technology called next-generation sequencing, or NGS. NGS can read millions of tiny DNA fragments at once, even if they're damaged or extremely short, making it ideal for ancient DNA. The sequencer works by attaching special markers to each base, allowing the machine to record the sequence of letters. Once this process is complete, scientists no longer have a handful of fragile molecules. They have raw genetic data, ready for analysis. Now it's time to understand what the DNA is telling us. Where in the genome does it belong? And more importantly, who did it belong to? To answer these questions, scientists use a step called alignment. Alignment is like solving a massive puzzle using a computer, and it is a difficult, time-consuming task. Each tiny DNA fragment, called a read, is compared to a reference genome, a complete, previously sequenced genome from a known species. The software searches for the best spot where each fragment might fit, a bit like figuring out where a torn page belongs in a familiar book. If a read lines up with regions known to be unique to Neanderthals, that's a strong clue. If it matches Denisovan mutations instead, it may have come from them. Once the read has a good match, the program aligns it to that location and logs the result. Each correctly placed read sharpens the picture, like adding pixels to a high-resolution image of our ancient past. But not all reads match easily. Some don't fit anywhere, or they look suspiciously modern. That's when scientists need to ask, is this ancient DNA, or is it contamination? To help answer this, scientists look for damage patterns that are typical of old DNA. One of the most important is cytosine deamination, a chemical change that often affects the ends of ancient DNA fragments, where cytosine, C, turns into uracil, U, and is later read as thymine, T. This kind of damage acts like a timestamp, showing that the DNA is truly ancient. Another telltale sign is the extremely short length of the fragments. Modern contamination is usually much longer. By spotting these patterns, scientists can tell which sequences are authentic and which ones don't belong. After scientists have identified ancient DNA from a particular hominin at a specific site, the big question becomes, What does it all mean? Who was here? When did they live? And how did they fit into the broader story of human evolution? To answer that, researchers turn to population analysis. This step helps them figure out which group of hominins the DNA belonged to, and how closely those individuals were related to each other or other known populations. Sometimes they can even estimate what percentage of the DNA in a sediment layer came from a certain hominin, offering clues about whether a site was occupied by a single group or shared by a mix of populations. They also combine this genetic data with other types of evidence: archaeological findings, environmental context, climate models, and dating techniques. DNA can also confirm, challenge, or refine what we thought we knew about a site's timeline and who lived there. Together, this layered approach can reveal when certain hominins passed through a region, how long they stayed, and whether they overlapped or lived there at different times, turning microscopic fragments into a rich narrative of ancient life. This is about more than discovery. It's about belonging. From a few grains of soil in a dark cave to full genomes of long-extinct species, sedimentary DNA is revolutionizing the way we tell the ancient story of us. Even without bones or tools, we can now trace where archaic humans walked, lived, and mingled, unlocking stories that history forgot, but the Earth remembers. As technology advances, we'll be able to detect even fainter signals, in even older layers, from even more mysterious ancestors. To push this revolution further, we are working on smarter field sampling methods and faster lab workflows, making it easier to recover ancient DNA and screen promising sediments more efficiently. The past is buried, but it's not gone. And with every layer we uncover, we get closer to understanding the whole story of who we are and where we came from. Follow for more insights into the fascinating history of us.